In this video we talk about the Megasporogenesis. Megasporogenesis. How the megaspores are generated, we have to discussing in this class. See the new cellulose, see here, this is the new cellulose. See here it is the new cellulose. In this the new cellulose, the small cell, here the one or more cells, they are converted into archisporial cell. Here the one or more cells of the new cellulose which is developed into archisporial cell, or which is converted into archisporial cell. Here these archisporial cell undergoes the divisions and forms a two cells, that is one is a sporogenous cells and another one is a parietal cell. See the one cell which is undergoes the divisions and form the two cells. In these two cells, one is a parietal cell and another one is a sporogenous cell. See here, these the parietal cell undergoes the periclinal divisions. Parietal cell undergoes the periclinal divisions and form the cells and that cells are added to this new cellulose. That cells are added to this new cellulose and the below the parietal cell, that is a megaspore mother cell. See here the sporogenous cell. See the sporogenous cell which is formed as a megaspore mother cell, which is formed as a megaspore mother cell. See here, this is a megaspore mother cell. Megaspore mother cell undergoes the meiotic divisions and forms the, here it is the diploid one, here it is the diploid one. Megaspore mother cell is the diploid one. That's why here these diploid megaspore mother cell undergoes the meiotic divisions and forms the four megaspores. That is this is called a megasporogenesis. See here the megaspore mother cell undergoes the meiotic division and forms the four haploid cells See here 1, 2, 3, 4. Here megaspore mother cell undergoes the meiotic division and form the four haploid cells. This is called the megasporogenesis. See here these four megaspores, these four haploid megaspores. See here in this picture, see here among these four haploid megaspores, here it is the micropylar end region, here it is the chalazal end region. Here the micropylar end region of the megaspores. See here the three megaspores, micropylar pole of these three megaspore cells. These are degenerate. These three are degenerate and the chalazal end, here the chalazal end of the cell, the chalazal end of the megaspore, this is a haploid megaspore which is act as a megaspore mother cell, which is in active condition. These three are the degenerate. Here only the active condition, it acts as megaspore mother cell and it acts as a megaspore mother cell and it forms the embryo sac. Which means here it is participated in the formation of the embryo sac. Here these are not the cells, these are the nucleus only. Here is the megaspore cell. Here is the megaspore cell undergoes the mitotic division and form the two nucleus, not the cell division, only the nuclear division. Here the two nucleus are there. It undergoes the mitotic division and forms the two nucleus. One nucleus goes to the micropylar region and another nucleus goes to the chalazal region, and these are undergoes the two sequential divisions. See here one nucleus undergoes the two, which means undergoes the one mitotic division and form the two nucleus, and it is also form the two nucleus and 2 or 4 nucleus, and here also 2 or 4 nucleus undergoes the mitotic division. These four nucleus are present at the micropylar region and another 4 nucleus are present at the chalazal region. See here it is the 8 nucleate stage. See here it is the 4 nucleate stage. See here the 2 nucleate, 4 nucleate and 8 nucleate. These three are undergoes the mitotic divisions only. Mitotic divisions of the nucleus. See, out of these 8 nucleus, three are undergoes the cell wall formation here. Undergoes the cell wall formation here, and see here it is the starting, and see the maturity embryo sac. See, out of these 8 nucleus, these micropolar end regions of these three nucleus are, out of these 3 nucleus, 2 are formed as synergids, and one is formed as egg cell. One is formed as egg cell, and another one is fourth cell is goes to the center position of the embryo sac, and it is called as polar nuclei. See here the downward, the chalagal end of the 4 nucleus. Out of 4 nucleus, 3 nucleus are converted into cell wall formation and formed as 3 cells, formed as antipodal cells. Formed 3 antipodal cells, and another fourth one goes to the central cell, and fourth nucleus, which is considered as a polar nuclei. See here, the synergids has the finger-like projections is there. So this is called filiform apparatus. See here, the function of this one is it absorbs the food material. They absorb and conduct the conduction of the food material from the nucellus to the embryo sac. And after the fertilization, these synergids and antipodal cells are degenerated. See here, the atypical angiosperm embryo sac contains 8 nucleate and 7 celled. It's a typical embryo sac. Typical angiosperm embryo sac has 8 nucleus and 7 celled. See here it is a 8 nucleus. 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleus but 7 celled. See here it is a 3 cell, here it is a 3 cell, and the central one, that is the big cell, that is the central cell, that is a 7 cell, but 8 nucleus, 7 cell. A typical angiosperm embryo sac contains 8 nucleus and 7 cells. This is the megasporogenesis. I hope you understood. Thank you.